Okay, hello everyone. Welcome to BIMAC Colloquium. I'm honored to welcome Professor Brian Delisle as our speaker today. Professor Delisle is a distinguished member of the Physiology Department and the Saha Cardiovascular Research Center and University of Kentucky College of Medicine. He had a PhD in physiology and biophysics from the University of Kentucky. And his research delved into the mechanism underlying cardiac arrhythmias, focusing on congenital long QT syndrome. And Professor Delisle's groundbreaking work explores the impact of molecular clock and circadian rhythm on arrhythmia susceptibility and aims to improve the treatment for the long QT syndrome-related arrhythmias. So please join me. Welcome to Professor Brian Delisle.
Yeah, thank you very much for that kind introduction. And today I'm going to talk a lot about the circadian regulation of cardiac excitability, but I do have a background in cardiac electrophysiology, and I touch a little bit on some modeling and things like that. Not so much in this presentation, but hopefully there's some work here that will stimulate people's interest in maybe modeling some of the things that we're showing, because it's always kind of fun to be able to work with people and understand data at a new level of detail and get new mechanism and ideas too. So before I get started today, I just want to really acknowledge all the people that contributed to this project. I'm first going to acknowledge Dr. Elizabeth Schroeder. I think she may be on listening, but so she and I have been working together off and on for a very long time. Since she was a postdoc and I was a graduate student. And so we've been fortunate enough to have a great career working together and diving into this field together roughly at the same time. And so that's been a real big pleasure of mine. Dr. Don Burgess, he does do a lot of our computational work and analysis. We've also been working together for quite a bit. Dr. Ono, he's did a lot of the work that I'm going to present earlier on in this presentation, along with Dr. Pravat. He's also contributed to some of the more in vivo studies that we've done. And last but not least, I definitely want to acknowledge Dr. Karen Esser, who's, again, a great friend, and we've been fortunate enough to work together and continue to work together in this area. And again, it's just such a great pleasure to be able to work with your friends on new data and new exciting topics. So I just want to first start off with a little bit of an introduction, and so just going to have an introduction talking a little bit about some of the background, what got me interested in circadian rhythms, and it was really this idea that there is a day-night rhythm in the incidence of sudden cardiac death. That's actually where I first started to become interested in these things. And then in the first part, I'm going to really start to dive in some of the mechanisms associated with the day-night rhythms in cardiac electrophysiology. And our group has really been interested in feeding behavior. We saw these observations about 10 years ago and are really trying to follow up on them now to get a little more mechanistic. But feeding behavior seems to be a pretty potent stimulus for driving some of the 24-hour rhythms in cardiac electrophysiology. And then in part two, I'm going to dive into the cellular side of things and talk a little bit about some of the work with the cardiomyocyte circadian clock, that transcription, translation, feedback mechanism that can drive gene expression. And we have some data suggesting that it does seem to regulate cardiac electrophysiology and ion channel expression, and then some newer data that may look at some of the conserved mechanisms that may be associated with the regulation of ion channel expression at the promoter level.
I see.
Okay. All right. So just a background. It's interesting. One of the things that I learned pretty quickly in studying circadian rhythms and that there is a pretty, depending on who you talk to, these words can have different meaning. And so for the presentation today, when I use the term circadian, that is going to be really referring to these internal biological clocks that are going to cycle with a periodicity of 24 hours. And similarly, circadian rhythms are these intrinsic physiological rhythms that will persist in the absence of environmental or behavioral cues. Okay, so these are true rhythms that are persisting in the absence of any external cues. But actually, most of the research that I'm going to be talking about today, I would classify as day-night rhythms, not necessarily circadian. They're not necessarily intrinsic, because what we're actually looking at is a combination of environmental, behavioral effects and how it's probably interacting with the circadian system. But only until the second part of the talk do we really start to dive into more circadian mechanisms. And so I'm going to be using the term day-night rhythms for the first part. Some people may use the term circadian to describe a 24-hour variation. If you do that, that's kind of what I mean by day-night rhythms, but it's really just a combination of behavior, environment, and internal rhythms. And so when we look at the incidence of sudden cardiac death, and this has been known for quite a while now, so really probably the mid-80s, I would say. And so just a little primer for people who may not study cardiology as a discipline. Sudden cardiac death actually has a pretty specific meaning too. So kind of just getting our vocabulary out of the way. It's the unexpected death due to an abrupt loss of heart function, and it's usually caused by underlying cardiovascular disease. Okay, so this is when people die suddenly, usually secondary to cardiovascular disease. It can be hard to identify in some cases, because you don't always know why someone passed away. So it is a little bit tricky. But we do have a pretty good grasp now on what's going on in terms of like the major causes for sudden cardiac death. So this is just kind of a little pie chart saying that the primary cause is coronary heart disease or ischemic heart disease. This is where you're kind of having issues with the perfusion of the myocardium. That's probably the leading cause. Cardiomyopathies are usually related to changes in heart structure. It could be acquired or congenital. Maybe you're born with it. And then genetic arrhythmia syndromes. That's actually my background primarily, and this is where you have genetic mutations in certain genes that predispose you to cardiac abnormalities related to electrophysiology. Usually the heart structure is pretty normal, but not in all cases. And so if you're just to look at the incidence of sudden cardiac death, you can kind of see here, this is actually time of day, okay? And this is a famous study by Mueller. And what they showed is that there is this morning rise, morning peak, if you will, in the incidence of sudden cardiac death. And I think most people think that this morning peak in the incidence correlates probably, with, ischemic disease. And so if you look at the incidence of myocardial infarctions, you see something very similar. You see this morning increase. Myocardial ischemia symptoms, again, you see this kind of peak in the morning that you have increased in percentage in the number of episodes here. And then cardiomyopathy is a little bit different. Like I said, this is where you have the structural change, and you also can see these morning increases in the incidence of sudden cardiac death. And so what's going on here? How do we understand this? And as someone who studies, sudden cardiac death, one of the major causes is usually going to be an arrhythmia. So you have cardiovascular disease, and something's culminated to the point where you've had a major arrhythmia, and that's what precedes the death. And to understand cardiac arrhythmias, we usually think of two things. You think of, an arrhythmogenic trigger, okay, some change that's going to trigger the myocardial substrate, that's the heart, to have a sustained arrhythmia that can result in death. Okay, so you almost have to balance this idea of arrhythmogenic triggers and how the substrate is responding to that trigger. Now, in healthy people, we're probably experiencing lots of arrhythmogenic triggers throughout the day, and there probably is this 24-hour variation in arrhythmogenic triggers, but most of our hearts are really healthy, meaning that our hearts are very, robust and protect against these triggers from culminating into any type of an arrhythmia that may kill us. Okay, and so that's the beauty of a healthy heart. It's very anti-arrhythmic, even if you expose it to certain arrhythmogenic triggers. The problem happens when you do have some heart disease. Okay, and so this is just your ischemic heart disease, for example. You change the myocardial substrate, and you lose that protective mechanism. And so now you can start to unmask these day-night rhythms and arrhythmogenic triggers. And then your arrhythmogenic risk can start to show a time of day pattern. And we think there might be something like this going on to manifest into this increased incidence of sudden cardiac death during the daytime.
So, Dennis, so that protective line is constant, or is it possible it has also circadian rhythm?
Yeah, no, this is a great question, and I think there's some there's some recent papers that would argue that it probably is not constant. But the short answer is I don't know. I think that's an active area of research. You can model it that way, and I think we did have a paper where we had like several scenarios. To me, I think the easiest way to understand it is, our hearts are pretty anti-arrhythmic. A healthy heart is really anti-arrhythmic. And that's where I think, you have this confluence of disease affecting the protection. And then I think that's where you start to see the reflection of the arrhythmogenic triggers. And there is quite a bit of data to show that they do have a pretty robust 24-hour rhythm. And so just to kind of, continue to answer that question, and please, if people have questions, please unmute and ask and things like that, because I don't have too many slides, and I'd much rather talk to everyone. This is just kind of our little schematic again of what day-night rhythms are. And there's obviously day-night rhythms in cardiac electrophysiology. So your heart rate and things like that go up and down across the 24-hour cycle. And a lot of people have worked to understand that daytime incidence in sudden cardiac death, showing, maybe it's attributable to blood pressure rhythms, making the heart work harder at certain times of day as your blood pressure is higher. Could be hormonal, could be neuronal. There's a lot of vascular changes. And like we said, that the most common causes are usually like related to ischemia and things like that. And there are time of day changes in how our, vascular tone is responding, endothelial function and platelets and things like that. So you do have this 24-hour rhythm in a lot of these things. And a lot of them actually do peak in the morning when you look at the physiology of them. And then, like I said, I kind of imagine that you add on some disease. And there's, again, a lot of mechanisms that you could imagine here, like a cardiomyopathy, ischemic disease, structural changes associated with heart failure, maybe fibrosis, genetic diseases like I was reviewing before. And so here you just have lost that protection, or maybe you've lost that protection at certain times of day and things like that. And then that can manifest into, these are cardiac action potentials, ventricular ones. And you get some irregular action potential behavior. And if that can get transmitted through the myocardium in a certain way, then it can promote these reentrant type of arrhythmias, such as that can lead to like ventricular fibrillation or some of these other types of deadly ventricular arrhythmias to manifest into this 24-hour rhythm. Or this day-night rhythm, I should say, in the incidence of sudden cardiac death, or in this case, arrest. And so we're really trying to understand this a little bit. And I think getting back to that question, are there day-night rhythms and protective mechanisms, and maybe, these different things? It's just a confluence of a lot of different factors. And especially if you look at different disease models, you can see differences in the incidence in terms of when you might expect to see sudden cardiac death risk increase. And so our first question was really looking at the mechanisms that are underlying the day-night rhythms in cardiac electrophysiology. And our model is going to be mice. Okay, we primarily work with mice. And we do have this hypothesis that feeding behavior is a major driver, really for the phase, maybe even a little bit to the amplitude in heart rate and ventricular repolarization. I'm going to kind of talk a little bit more about that in a second. And so just a reminder, I know most people are probably familiar with an ECG, but ECGs in mice are a little bit different. And so we have the same kind of setup. You'll have atrial depolarization, and that'll be conducted into the interventricular septal wall here via bundles of His, and then out along the Purkinje fibers on the ventricular walls here. And that depolarizing wave from the atria to the ventricles, and then even the repolarizing wave, we can visualize that by recording the electrical activity of the heart on an ECG. And so this is just looking at a cardiac cycle, one single heartbeat. in terms of the electrical activity in a mouse. And what you can see here is the P wave, QRS, and there's a J wave and then a T wave here, and it's actually inverted compared to people. And here, we're going to be primarily looking at the R to R interval, and that's a surrogate for heart rate, essentially. So you can really use those terms interchangeably for the purpose of this presentation. And then here we have the QT interval. The QT interval is going to be a measure of depolarization and repolarization. And so we are very interested in the QT interval because a lot of research shows that if it's too long, that can be proarrhythmic. If it's too short, that can be proarrhythmic. So it's just a really nice indicator of some of the changes that you may see. And then just to kind of talk a little bit about the J wave, because that's not something you normally see on people's ECG. So P correlates with atrial depolarization. QRS is ventricular depolarization. The mouse heart, it has to beat usually 500 to 600 times a minute. And so it will repolarize very quickly. So they have a very large early repolarization phase, and that's what culminates into a J wave. And then you do see a little bit of a slower repolarization, and that gets your T wave as well. Okay, so I think, yeah. Okay, so we have a premise here. So why do we think feeding is so important? And like I said, you know, probably about 10 years ago now, we were looking at this, and we were really interested in understanding, you know, different kinds of Zeitgebers or timekeeping cues that may be important for physiology and the heart and things like that. And feeding behavior is thought to be a really important timekeeping cue that may be regulating day-night rhythms. And so we did some study again in mice using telemetry to record the ECGs, and this allows us to continually record them in conscious free moving mice. And mice normally eat most of their food during the dark cycle. And so probably about 80% of their calories during the dark cycle. And so what we would do is kind of force them to eat during the light cycle through a time restricted feeding protocol. It's not caloric, it's time restricted feeding. And so from there, you know, what we can do is measure the impact of this light cycle restricted feeding on the cardiac electrophysiology. And just let me walk you through this. So here is our ad libitum fed mice in the black circles here. And this is now looking at the RR interval. And you can see it cycles maybe between about 100 and, you know, 10 to 120 milliseconds across the course of several days here. Notice I have Zeitgeber time. Zeitgeber time 0 just means the lights have turned on. So every time you see a 0, the lights are turned on, and these are 12-hour light dark cycles, and that's what's indicated by the shading. That would be a dark cycle. And so what you can see here is that normally the mice have a really long RR interval during the light cycle, which is what you would expect. That corresponds to a slower heart rate when they're sleeping. But when we did this light cycle restricted feeding for a couple weeks, we did it for about 2 weeks, we saw some really dramatic changes in the RR interval, and that's highlighted here in the blue. And notice it got a lot slower, and then notice the peak. And so here it's peaking during the ad libitum fed. Here it's now peaking during the light cycle restricted fed condition. And it really did shift quite a bit here. Okay, and now it's actually slowest during the dark cycle. And so that really kind of caught our attention. And we also noticed that QT interval, that interval associated with depolarization and repolarization, had a corresponding change, you know, so it kind of mirrored what we were seeing with the RR interval, which surprised us a little bit because there is some data that in mice in particular, they may be independent of one another. Okay, so now we're going to, you know, kind of revisit this study, and we're going to do it again, and we're really going to try to understand some of the mechanisms that may be causing these changes in the 24-hour rhythm of the heart rate and ventricular, you know, depolarization and repolarization. And so here's our setup. Again, continuous telemetry monitoring. So free-moving animals with little implants that allow us to record the electrocardiogram. We're going to be recording core body temperature. I'm definitely interested in core body temperature rhythms. And then We also record activity. I'm not reporting that data, but basically the activity rhythms really didn't change too much. We were seeing large changes in the ECG and core body temperature. So I'm going to focus on that.
Mm-hmm.
And so what we see here is our setup, 12-hour light dark cycles, and then ad libitum fed mice mostly eat during the dark cycle. And then we're going to time restrict their feeding to the light cycle, not caloric restricted. They do eat about the same amount, especially after the first day or two, as ad libitum fed mice. And so again, this is kind of looking at the data. So here we are looking at the ad libitum fed mice. These are some ECG traces that we generated using ensemble averaging. Kind of helps us to measure, especially the QT interval. That can be pretty challenging. And we're just looking at hourly average data here of the RR interval, and here's the QT interval. We also measured the PR interval. That's a measure of conduction between the atria and the ventricles. I'm not going to talk too much about that. The rhythms were pretty small, and it was kind of hard for us to kind of follow up on it. But what we saw is it kind of mirrored what we were seeing with some of these other ECG changes. And so here we have our, ad libitum fed light dark cycle, and we're seeing a nice peaking of the RR interval in the light cycle, again, suggesting slow heart during the daytime, faster heart rate during the nighttime, which is what you would expect with the mouse. Now, in this study, we just actually started to really look at the data pretty much 1 day after doing the switch to the light cycle restricted feeding. And so that's what this means, this initial here. And this is always amazing to me, but 24 hours after starting this protocol, look what happens. Boom, it's like you generate a new rhythm. And so here's your kind of old rhythm. These mice have been light cycle restricted feeding for just a day. And then all of a sudden we have this new rhythm now where the heart rate is now peaking in the dark cycle, and then, fastest during the light cycle when they're now eating. And we see corresponding changes in the QT interval. And these are pretty large changes, I would say, in the QT interval. And then when we continued this protocol for 2 weeks, we saw pretty much that same rhythm. It might have adjusted a little bit, but we saw the major change within right after about 24 hours. And then it kind of settled into this new rhythm, and it didn't change much, as we continued it for several weeks. And so one of the things that we did this time was really to look at the core body temperature changes. And, core body temperature, that's going to mirror what you would expect to see in the RR and QT interval. And that just means that the lower the core temperature, the slower the heart rate, or the longer the QT interval. And so we did see some large changes in the core body temperature also. Okay, and that was pretty interesting to us. It wasn't too unexpected. This actually has been published in some of those earlier studies, in particular, that we're looking at, what kind of cues could align, day-night rhythms and different levels of physiology. And so this is something that people have seen before. And so it does seem like we're mirroring what we saw with the ECGs. And so we're going to kind of explore this in a little bit more detail. And
so, I'm sorry. So, do you think this R interval change and body temperature change are just parallel, or one is a cause and another is effect? Do you?
Yeah, no, it's a great question, and this is actually where we're going. So it's
I
a
see, I
little
see.
bit
You
of
will
both,
not.
I think.
Oh,
Yeah,
yes. Okay.
yeah.
Great. Thank you.
Yeah. No, this is kind of this is where our thoughts are. I think the initial way we kind of did this is like, how well do they correlate, right? Associate, but that's not causation. And so let's do an experiment, and look a little bit more if we can see how much of this is related to temperature, really. And so this is, again, one of the things that we like to do is these cosigner wave fits. So we basically make sure the data is rhythmic. So we do a statistic on the individual mouse level. Okay, so we'll actually go to each individual mouse, fit the data, do a statistic to ask if it's cyclical. And if it does pass that first test, that significance test, then we do the cosine wave fit. And then we can drive the parameters from the cosine fit. Like acrophase, that's when we get the peak of the rhythm in terms of the ZT. The time of day, relative to the start of the light cycle, the average 24-hour rhythm, and then the amplitude of the rhythm. And there's a lot of data here. I don't mean to kind of show a lot of data on one slide, but basically it's kind of the same data, just looking at the RR interval, QT interval, and core body temperature. And you just notice that those 24-hour rhythms, if you just quantify them, they're kind of doing the same thing. Peaking during the light cycle with our ad libitum fed, and then peaking during the dark cycle as soon as we 24 hours after starting that light cycle restricted feeding. And it still pretty much stays around that same peak, if you will, even after 2 weeks. So it does seem like all of a sudden we're just getting this new 24-hour rhythm, if you will. The average, or the 24-hour adjusted rhythm, the MESR, also increased. So the average is a little bit slower. And that probably is related to this amplitude increase. We do see a large increase in the amplitude of both the RR interval, saw similar changes in the QT interval as well. And remember, the changes in the body temperature are more or less mirroring what we see with the RR and QT interval. So normally it peaks during the dark cycle, but when we did our light cycle restricted feeding, it's now peaking during the light cycle. And then we saw kind of like these converse change in the MESR. So as you cooled off, the intervals got slower. And then we did see a parallel with the amplitudes, which is actually what you'd expect as well. So it does seem like these things are kind of correlating in terms of how their 24-hour rhythms are changing. So now we did do some estimates on how much the heart rate would change as a function of core body temperature. And you can kind of do a couple back of the envelope calculations using Q10 values. Some are reported in the literature. And there are some really cool papers that have been published recently on this. And so what we did is we just took the MESR, adjusted for a Q10. In our case, we did 2.5. And that was based on a recent paper looking at an ion channel that regulates heart rate, the HCN channel. And when they looked at the whole heart, they were seeing about changes in the isolated heart as a function of temperature. And they were getting about 2.5 Q10 values. And so we just kind of estimated the change in the heart rate just based on the changes in core body temperature. And that's actually what's being highlighted in green here. And then gray is the RR interval data. And actually, we were a little surprised, but during ad libitum fed conditions, they go along pretty closely together, but there are differences if you zoom in a little bit. But here's where we actually start to see a divergence. So once we get into the light cycle restricted feeding, particularly after the first 24 hours, you notice that the estimated change in the heart rate, or the RR interval, based on temperature, did not seem to completely explain all of the data we were seeing. So just using this estimate, it looked like there was something else going on here that may be explaining the changes in the RR interval with the light cycle restricted feeding. And so that prompted us to look at heart rate variability analysis. And so I don't know if anyone's familiar with heart rate variability analysis. It is a field into itself. We have studied it quite a bit. We mostly focus on power analysis. So kind of looking at frequency domain components. People will look at high frequency and low frequency, and it's been worked out in mice, what ranges to look at. And just to kind of give a primer, I would say the high frequency component is generally accepted as a surrogate for parasympathetic outflow, okay, to the heart, okay? And probably what's happening is as we breathe in and breathe out, our heart rate adjusts with that. There's a little bit of parasympathetic activity with each breath, and we call it the respiratory sinus arrhythmia. And what it is there is some signaling there associated with parasympathetic activity, and that's probably what we're capturing in the high frequency domain. So I think most people would accept that's probably what we're seeing with the higher frequency component. You'll often see low frequency data reported. We're not going to report that today. That's a lot more complicated, I think, especially in mice. it's probably a combination of multiple components. So we're just going to just focus on the high frequency. And it looks like we're getting a slowing of the heart rate, which you probably expect to see more of that parasympathetic signaling going on. So we're just going to kind of focus on that. And when you do, you notice that the power of the high frequency component did seem to increase quite a bit, corresponding to the times where we were seeing a divergence between the estimated heart rate due to body temperature and the actual measured heart rate. And so it looks like we're starting to see now correlation here with changes in the high frequency component, RR interval, and that may be contributing to this extra increase in the RR interval.
So
Then, go ahead.
green is experimentally measured one, and the gray
The
dot.
other way.
Oh, so
Gray
is experimentally measured,
Yeah.
and then a green dot is estimated from the body temperature. So
Yeah.
how can you estimate the one from the body temperature? So based on some correlation thing, or
Yeah, so basically we just take the Q10 value and then the average core body temperature, and you can do a little back of the envelope calculation to estimate
I
it.
see. You do
And that's what I meant,
regression.
like, yeah, we
Okay, I
used, if
see.
you're looking at heart rate, our Q10 was like 2.5,
Okay,
and I
gotcha,
think
gotcha.
I've seen it, I think 2 is, one, and then like I've seen as high as 3, and I've seen as low as like 1.5. So it's just kind of a way to get an idea of how much the heart rate change may be attributed. How you define the mean is probably a good conversation to have. Like what are you going to use as your mean? I think that's a great
I
conversation.
see, I see. Wow.
We just did the 24-hour average, so.
Okay, I see. So using the body temperature, you estimate the RR interval, that's the green curve, but that cannot fully explain the observed RR interval change.
Right. And that's what prompted us to do the high frequency analysis.
Wow.
And it looks like we're seeing increases in the power of the high frequency component that corresponds to where we start to see the divergence here between estimated change in body temperature, heart rate associated with body temperature and measured. And then, so building on this, we're like, well, we actually had trouble fitting these data to any kind of, cyclical analysis. Okay, so we couldn't really use cosine fitting here. And so we used a slightly different approach to see how well these two variables were correlating. And so now we're focusing on measured heart rate and the power, associated with the high frequency component of heart rate variability. And so this is an arrhythmic, it's a type of rhythmic analysis. And so we're really looking at the correlation coefficient between these two variables. And what we're seeing here is that, they're basically lagged against each other. What we see with these correlation data is they peak together at lag 0, meaning that, these rhythms are probably peaking together in time at the resolution of 1 hour, okay? And so they have a pretty strong correlation, and notice that even when we do our light cycle restricted feeding, it's pretty similar. It looks like they're kind of, moving along together as well. And then the correlation stays pretty high. Data, I'm not showing activity. We saw actually a pretty strong correlation in ad libitum fed, but we actually lost it when we went into the light cycle restricted feeding. So we don't think a lot of these changes are associated with changes in activity. And then, so these are all correlations. So let's get into some experiments. Let's test this idea, right? So is this related to autonomic signaling, like the heart rate variability analysis suggested? And so when we did this, what we decided to do was to do some pharmacological experiments using different drugs, propranolol and atropine. So propranolol is a beta adrenergic receptor inhibitor, and atropine is a muscarinic receptor inhibitor. And so they'll inhibit the activation of the beta and muscarinic receptors. And so this is analogous almost to like autonomic block. We're just kind of blocking the autonomic input to the heart, both sympathetic and parasympathetic. And we're going to ask, when we do that, what happens now to the change in the heart rate, particularly during that light cycle restricted feeding, where we're seeing these really large rhythms that are kind of in a very different phase than you would expect, peaking during the dark cycle. And to do this, since we were kind of more or less interested in the 24-hour component, we're going to do repeated injections. Okay, so we're going to inject really where the rhythm's going to be, the heart rate rhythm is getting faster, and then where it was expected to be getting slower. Okay, and so this LC, that just means light cycle 0, dark cycle 0, light cycle 1. Now we're starting our study, dark cycle 1, light cycle 2, and dark cycle 2. And these arrows are corresponding to the time of day that we're doing the injections. Okay. And so when we do this, what you can see is that pretty much after starting the injections, the heart rate, and we're also doing the estimated heart rate as a function of the core body temperature, pretty much goes flat here. Okay. And so again, kind of suggesting to us that, well, we're inhibiting the autonomic nervous system. It looks like we're losing our rhythm in the heart rate, even during that light cycle restricted feeding. And so we can quantify it using day-night now. So now we're just looking at the average heart rate during the light cycle and dark cycle. And, you know, when we're doing the light cycle restricted feeding in the absence of autonomic blockade, we see a very nice difference. Again, it's slower in the dark cycle, which is a little unusual, but, you know, it's a lot slower here. But then after doing the autonomic blockade, this would be day 1, light cycle, dark cycle, day 2, light cycle, dark cycle. We just don't see that difference anymore. And so this really started to suggest to us that with light cycle restricted feeding, some of these changes that we're seeing in the heart rate in particular are probably related in part to the core body temperature changes as well as autonomic changes.
I see.
And so now we wanted to focus on the QT interval, and we're really interested in the QT interval just because it's one of those things where you know, in mice, the heart rate and QT interval are thought to be regulated by slightly different mechanisms. Okay, in humans, actually, they are dependent on each other, and we actually have correction formulas for as the heart rate changes, the QT interval changes, and we can correct for that. But in mice, there's a really weak dependence of the QT interval duration on the heart rate. And so we were looking more closely now at the changes in the QT interval with body temperature. And there were some previous studies in canine models where they were looking at linear relations between the QT interval and core body temperature. They were focused on heart rate corrected QT intervals because it does change with heart rate in larger mammals, but since it doesn't really change that much with heart rate in small mice, we figured we'll just try to do the linear correction, you know, linear, look at the linear relation directly. And so this is looking at each mouse now, their QT interval under the different ALF and light cycle restricted feeding plotted as a function of the core body temperature, and just doing linear analysis, just, you know, straight up linear analysis. And it looked like we got pretty decent, you know, correlate R squared value, I should say. And the slope value was about, you know, maybe 4 to 5 milliseconds per every degree. Okay. And one trick that people use just to kind of say, okay, you know, can we now like correct, if you will, for the QT interval for a certain temperature? And so we worked on that a little bit by incorporating the slope factor, did it for 37 degrees. And so now we've, you know, kind of corrected for the temperature dependence, if you will. And then you get a nice, pretty flat line, I would say. And so that suggested to us that, okay, we can correct for the change in the temperature and get a pretty reasonable stationary QT interval. And so what we wanted to do then is look at our data. And again, the gray is going to be the plotted measured data here. Okay. And so that's what we measured on a previous figure where the QT interval was changing quite a bit with the light cycle restricted feeding. And now the blue is just kind of that corrected QT interval. So we've now kind of corrected it, you know, for a temperature and kind of, you know, 37 degrees in this case, and said, okay, well, what if the QT wasn't changing as a function of temperature, essentially? And now you can kind of see that we have really flattened out this day-night rhythm in the QT interval. And so that suggested to us that maybe a lot of the changes that we see in the QT interval were primarily reflecting what we were seeing with the core body temperature. And so maybe they're a little bit more related than what we were seeing with the RR interval. And so this is kind of a tricky thing to separate, right? We're like, okay, so how are we going to separate this? Because it looks like the day-night rhythm and the RR intervals, depending on, both the core body temperature and the heart rate,
Yes.
or excuse me, the autonomic nervous system.
Uh-huh.
And then the QT interval, we think it's going to be primarily reflecting the core body temperature. And so again, so how can we maybe change the heart rate through autonomic regulation without having large changes in the core body temperature? Okay, and that might help us to separate, if we really are seeing distinct mechanisms that are regulating the day-night rhythms in these variables.
Wow.
And so what we decided to do, there is a rich literature on this, is to house the mice in thermoneutrality. Okay, and this is just now back to standard ad libitum fed. We're not doing light cycle restricted feeding because our question is very specific.
Mm-hmm.
If we put them in thermoneutrality, studies have shown that the mouse metabolic rate will slow quite a bit, and their heart rates will slow quite a bit. And it's really due to, it's thought to be due to an increase in parasympathetic drive to the heart. Okay, and so the idea is that when you put them in thermoneutrality, they do resemble more like what we would expect to see in people, where the parasympathetic tone may be a little bit higher, heart rate's going to be a little bit slower. And so we just asked the question, let's stick them in thermoneutrality. It's a drug-free way to augment parasympathetic signaling to the heart. And then let's just measure the corresponding changes in the RR interval and the QT interval. And we have our little estimated, RR interval here in green now. Okay, and then we have the QT interval. And again, this is ad libitum fed conditions. So this is not time restricted fed. And you can see, we do see a really pronounced slowing of the heart rate. It's now peaking in the light cycle, which again is what you'd expect with ad libitum fed because these mice are probably inactive and not eating during the light cycle. And so we see, oh yeah, the 24-hour rhythm and heart rate really is influenced by the autonomic nervous system. We can't really explain or account for it simply just by changes in core body temperature. Okay. Now, when we look at the QT interval, and so we're looking at, we could actually still pretty much correct for this day-night rhythm in the QT interval, just using our same formula that we derived for the mice measured at room temperature. And so it seemed to us that we're seeing large changes in the RR interval, and we think it's really related to changes in the autonomic input to the heart, specifically probably parasympathetic. But again, we're kind of not seeing these dramatic changes in the QT interval, and we can pretty much correct it using the same formula that we did at room temperature. And so we did similar analysis. Let's go ahead and look at the slope now. The temperature range is a lot less, right? Because the temperature, they don't, cool off quite as much as they did in the light cycle restricted feeding. But we were still able to kind of resolve slope analysis here. And when you looked at the slope, it was pretty similar to what we saw in mice at room temperature. And again, we could correct for it pretty well, suggesting to us at least, it looked like the QT interval, again, was still primarily, the day-night rhythm in the QT interval was still primarily reflecting core body temperature, even in mice housed in thermoneutrality. And so this is just kind of a summary slide of everything I just kind of talked about. So I'm not going to repeat it too much. I would like to highlight some of the key points here. It does look like, we get similar changes in the day, night, and 24-hour rhythms in the RR and QT intervals and core body temperature. However, we think the changes in the heart rate are reflecting a combination of core body temperature and autonomic input, where QT is primarily reflecting core body temperature. And to us, this is suggesting that the day-night rhythms in cardiac electrophysiology are not really aligning to light-dark cycles, but rather maybe the feeding cycles. And so that, I think we're kind of starting to see that coming out here. And then maybe heart rate and ventricular repolarization are aligning by distinct mechanisms, meaning that one is going to be more dependent on maybe autonomic changes. The other right now seems to us to be more dependent on core body temperature rhythms.
Wow.
Okay. So I can go into part 2 a little bit here, and I'm going to take a big shift. And I'd like to start to talk a little bit about cellular rhythms. And now I'm going to start talking about the circadian clock mechanism. And I know most of y'all are probably familiar with the circadian clock mechanism, but just as a primer and to review, this is that transcription translation feedback loop where we have the transcription factors BMAL and clock representing a positive limb of the circadian cycle where they're going to drive the expression of period and cryptochrome, which can negatively feed back on the activity of period and clock. And due to the degradation of period and cryptochrome, you can establish a periodicity in this transcription translation feedback loop of about 24 hours. Now, in addition to this core loop, these transcription factors and maybe even some other components of the clock that's not shown here, this is a very simplified diagram, can regulate the expression of a lot of other genes. Okay, and so there may be direct interactions. They may be regulating the expression of transcription factors, which in turn regulate the expression of other genes. So this is a network, if you will. And so one of the things that we were really interested in is how much of this core clock mechanism, or maybe some of these downstream clock controlled genes, are regulating cardiac electrophysiology. Okay, and I'll kind of go into that a little bit, and then talk a little bit about some of the newer stuff, maybe if we have time, about the regulation of ion channel gene expression. Now, some of the earlier studies looking at the core clock mechanism would delete BMAL in the whole animal, or just in the cardiomyocytes, from the moment of early on, like these are knockout animals, okay, and so the germline knockout has a lot of systemic issues. It doesn't have circadian clocks in any of the cells throughout the body. One of the things that will develop is dilated cardiomyopathy and premature mortality. Okay, and so that's all this is saying, is that the germline knockout a BMAL, that core clock transcription factor, really makes the animal sick. But there is a cardiac phenotype, which is kind of interesting. And then another kind of model, if you will, is the cardiac specific, cardiomyocyte specific BMAL knockout. And when you look at that, what you'll actually see is kind of a similar phenotype as the germline. They actually do develop a dilated cardiomyopathy and premature mortality. Now, we were more or less interested in trying to actually understand it in the adult myocardium. So there can be developmental issues and things like that. And so what we did, and this is really work with Dr. Karen Esser, was looking at the deletion of the BMAL. So now we're using an inducible kind of system where we can actually induce the deletion of BMAL in the adult heart. So you let the mice grow up, and then you inject them with tamoxifen, which allows for a recombination of the BMAL allele, and essentially creates a deletion, if you will, of the BMAL. And we did that in the adult heart. And this is just looking at qPCR of BMAL and DBP. And we saw, really reduced rhythms. Some people kind of point out, wow, they didn't go away. But you got to remember, the heart is not just cardiomyocytes. It's actually about 60% other cells. And so we actually think this is a pretty good signal that we disrupted the cardiomyocyte circadian clock. And then kind of looking at the hearts, we didn't see that dilated cardiomyopathy. And we've actually aged these animals out quite a bit. We still have yet to see functional defects, and we still have animals growing old in our colony, but we haven't really seen any structural deficits. And we perform like echocardiographs where you can actually look at changes in the structure and function. The left ventricular mass is not different in these animals, so we're not seeing any kind of hypertrophy or dilation. And the ejection fraction is just a fractional amount of blood that's ejected with each beat. Seems to be the same, so we're not really seeing any mechanical dysfunction as well. And so what we did see, though, was an electrophysiological phenotype, and that's what got us really into working with these mice, is that, structurally normal, electrophysiologically not so. And what do I mean by that? They still had beautiful day-night rhythms in heart rate and the QT interval, but the QT interval was just longer. Really. I mean, I think that was the main thing that we saw. It just on average was longer. And what we kind of estimated from this is that maybe we're getting changes in ion channel expression. Maybe that circadian clock, either directly or indirectly, is contributing to the expression of ion channels that may be impacting the repolarization properties of the heart.
Mhm.
And so what we wanted to do, and this is kind of summarizing a lot of work, is to just look at, again, using qPCR in this approach, just different cardiac ion channel genes that are known to be important for mouse cardiac electrophysiology. And so here again is our ECG, cartoon of it, PQRSJT. This is really the corresponding mouse ventricular action potential. Remember I said that early repolarization really drives that J wave, if you will. And the action potential is just a reflection of the inward and outward currents,
Mhm.
in the cell. And so when you kind of take a look at it, there's quite a few here that are in the mouse, a lot of repolarizing currents. And you can look at the different genes that will encode the proteins that contribute to these ionic fluxes across the membrane. This is not all of the genes, but it kind of highlights some of the patterns I think that we saw. In black here would be the control animals, and red was our tamoxifen injected or induced B-mal deletion animals. We generally would see a reduction in transcript levels.
Mhm.
Some of the reductions occurred in transcripts that looked to be rhythmic. Maybe they did have a circadian rhythm, and we could do some statistic to
Mhm.
test for that. But we would even see reductions in some transcripts that didn't have an obvious rhythm. So it suggested to us that inducing the deletion of B-mal not only maybe disrupted some of the rhythmic expression of certain ion channel genes, but it may also affected the overall expression of ion channel genes. Again, maybe through direct or indirect mechanisms.
Mhm.
And highlighted here in red is all the ones that we did see some changes with. Not all of them are shown here, but most of this work has already been published. Now, one of the ones that really fascinated us was this KCNH2. Okay, and so why is that? One that had the most beautiful rhythm. It's one of these that has, like, every database you look in, it has a very beautiful 24-hour rhythm and transcript
Mhm.
levels. Again, this is actually done in constant darkness. I didn't say that. So, like, when you're doing constant conditions, you can still see it. It's really nice looking. And then one of the weird things, though, is actually when you look at the mouse,
It
it
doesn't.
really doesn't contribute to the action potential. So it's probably not explaining some of those QT changes.
Oh.
But it's one of these channels that is very interesting to me. I'm actually a biophysicist, and it has
Yeah.
very unique gating properties.
Yes.
And so here's again, just the same data. Here's our control animal, beautiful 24-hour rhythm and
Mhm.
KCNH2 transcript levels. Inducing the deletion of B-mal seem to disrupt this rhythm. And we can measure the functionality, even though this channel is probably not contributing to mouse cardiac electrophysiology, it's still expressed. It's still there. And so we have to do some tricks, if you will, to kind of isolate the currents from all the other potassium currents. It's very difficult to measure because it's very small. But we were able to kind of resolve it using some biophysical techniques. And it looked like after inducing the deletion of B-mal, it was quite a bit smaller. And so that was pretty interesting to us. And why are we so interested in this current is that it's been linked in humans to a lot of arrhythmias. And what do I mean by that? A lot of medications, a lot of drugs in development and things like
Mhm.
that will block this current.
Mhm.
There are mutations in the gene that will reduce the current, and that they can prolong the ventricular action potential and predispose people to a polymorphic ventricular tachycardia and sudden cardiac death. And so it just seemed to be a very interesting
Mhm.
channel for us to kind of follow up in. I know I'm running a little bit long
Yeah.
here, so I'm just going to summarize a few slides
Mhm.
fairly quickly. And again, some of this data is already published, but
Mhm.
we wanted to just test the idea that, whether or not this gene, some of the mechanisms associated with its circadian regulation may be conserved. So going from mouse to human. And when you look at this gene, there's actually two major transcripts. There's the full length and a shorter transcript. Both are in mice, both are in humans.
Mhm.
And when we actually looked at the 5' upstream regions of these transcripts, and they have alternative start sites, so it's kind of a unique setup. It's not just alternatively spliced. They actually have different 5', different first exons, if you will. Really, we saw a lot of conservation between the mouse and the human in the longer form, this casein H2A, which is really the one that's probably the major functional one in humans. And so this is just looking at promoter homology, and we saw a lot of conservation there. And what we're doing now, and I'm probably going to just close on this, is doing some expression studies looking at promoter reporter constructs where we can actually use the promoter from the casein H2 to drive a reporter luciferase and then measure its the circadian activity. And you can do this using this technique LumiCycle. And so basically you can culture cells expressing the promoter of these genes driving luciferase, and you can then measure their circadian activity. And right now, we've actually, I'm just going to talk about this briefly. Dr. Schroeder is the one that developed this technique in our lab, but it's based on some published data. What's really cool is to synchronize the cells in vitro can be a trick, and their circadian clock mechanism. Yeah. And we're using the tissue culture temperature. You can cycle that, and that will actually align the clocks in the cells. And that's actually being shown here, looking at luciferase activity of BMAL and period, how they're antiphase in our in vitro system. And so I'm just going to kind of end there. What we're doing now is basically looking at the casein H2 promoter and looking at what regions may be important for its circadian expression. And it looks like we may have identified some E-box elements and things like that, where you may expect some transcription factors to bind that may be important for it. Okay, but just real quick, I'm just going to give some acknowledgements. Again, I gave some in the beginning. Some other people I'd like to acknowledge: Rebecca, Andrew, Sydney, and Tanya. And then obviously Karen Esser and Jiping has been working with us quite a bit too. So, and obviously I'd like to thank you all. I really do appreciate the opportunity to talk to you. And I apologize. I went.
No, it's okay. Totally fine.
Sorry about that.
No, thanks so much. Okay.
Yeah, this is fun.
Yeah, let me stop it.